The front part and the biggest portion of the brain is the forebrain, which is the front portion and the anterior portion of the brain. It is also called in medical terms as prosencephalon. So there are three main portions in the forebrain, which are namely the olfactory lobe, second is the cerebral hemisphere, and the third is the diencephalon. So when we see this diagram again from the NCERT, when we look at the bottom, see this is the side view of the brain. When we turn this brain in this manner, so when we try to see the lower portion of the brain, we can easily see there are two small portions which are the olfactory lobes, and these portions are the cerebral hemispheres, and then the third portion is the thalamus, which is not supposed to be visible in a normal diagram. So when we talk about the olfactory lobe, the first portion, which is also called in medical terms as the rhinencephalon. So rhino word is used for nose. And we all have learned in 10th class, olfactory or olfaction is a property of smell. So olfactory lobes are the lobes which are a pair. There is one pair of structure present towards the lower surface. That's why it is called here as inferior surface or the ventral surface to the hemispheres, and these lobes carry a cavity. That cavity is called as the rhinocele. Cele word is always used in biology for a cavity. You'll be learning this name again and again in the first unit also. That means the animal kingdom. So, but remember, this rhinocele is absent in the case of human. So in the case of human, olfactory lobes are poorly developed. That's why if you compare a human smelling power with the dog or a shark, we all know human is not able to smell nicely. We don't have a strong olfactory power because our olfactory lobes have become shrunk in size. So we are more of thinking type of brain carrying individual instead of smelling. That's why, because this cavity is absent in the case of human, otherwise this cavity is present in the other animals, and it is called as the first cavity present in the brain. It is the first ventricle. Ventricle stands for cavity in the lower animals, and it is the seat of smell. In human, it has a tract and a bulb. So olfactory lobe consists of a tract, a long rectangular portion, and then a bulb is there. So there are one pair of this structure is present, and there is no cavity, there is no smelling power. So it is a part of, so it is only important in the form of a pathway. So it's a pathway through which the stimulus of sensation of smell goes to the temporal lobe. We'll be talking about temporal lobe within few minutes. So brain has a portion which is called as the temporal lobe, and temporal lobe is the lobe in which our smelling power is residing. So in the case of human, olfactory lobes are functionless. They do not have the property of understanding the smell, understanding the flavor or aroma. It only has the property of passing on the stimulus to the main portion of the brain, that is the temporal lobe of the brain. The next portion of the brain is the cerebral hemispheres, which are called as the telencephalon. They are the largest part of the brain, and you can say largest part of the forebrain also. They constitute 80% of the volume of the whole brain, and cerebrum is not just one structure, it is a pair of structure. So there are two small hemispheres. It is something like when we break a walnut, we find structures like this. So in the same manner, there are two lobes which are called here as the two hemispheres, separated by a deep longitudinal fissure. Fissure is a depression in biology, and then both of them are attached with the help of a body which is called as the corpus callosum. Corpus callosum is 10 cm long curved band which is made up of around 200 million myelinated nerve fibers. So this is the corpus callosum, and this corpus callosum has a curved portion present here and a curved portion present here, which are called as genu and the splenium, and then there are two knob-like structures present on both sides which are called as the fornix. Both hemispheres convex on the superior side. So when you pay attention to the brain, you can see that there is a convex surface here, a concave surface here, and this portion is the corpus callosum. You can see a depression here, a front portion is curved, and again a knob-like structure present here. So the both hemispheres are convex on their superior side, and then they are concave towards the lower side, and they are flat on their median side, and left side is supposed to control the right side, and right side is supposed to control the left side of the body. This is the concept is of brain, that brain always has the left side of brain controls the right side of our body, and in the same manner the right side controls the left side of the body. The next part is that the cerebrum is divisible into two parts. The outer portion is called as the cortex, which is grey matter, again same terminology, and then the inner portion is the white matter, and it is called as the medulla. Now the grey matter carries main centers as per the NCERT. It has the center of sensory, so sensory area, motor area, and association area, which I just read few minutes back from the NCERT book. And then the white matter is rich in myelinated nerve fibers. Now there are cavities or ventricles present in the case of the cerebral hemispheres also, because there are two cerebral hemispheres. That's why there are two cavities there. So each cerebral hemisphere carry an irregular cavity which is called as the lateral ventricles. Remember, rhinocele or the cavity of the olfactory lobe is absent in the case of humans. So humans' first cavities are the cavities present in the lateral ventricles, which are present in the cerebral hemispheres. These are called in biology as paracele. So that one was rhinocele. These are called as the paracele, first as well as second, because two are in number, and they are lined with ependymal cell. If you connect this with your 7th chapter, I have discussed these cells in the 7th chapter, that these are epithelial cells, cuboidal in shape, and they carry cilia. And the reason is because these cavities are filled with cerebrospinal fluid. That fluid has to be moved, so that motion is generated with the help of the cilia present on the surface of ependymal cells.